Well, welcome back, everybody. We've got an exciting session planned for you today. Remember to join us on Twitter. Don't forget the hashtag is hashtag 2022 IEP Workshop, all one word. And that'll help us continue this conversation both during the conference or the workshop and after the workshop. So again, hashtag 2022 IEP Workshop. We've got 16 or 17 lightning talks for you. We're just going to roll through them. Very quick introduction. And then we'll move into the poetry slam, and we'll take whoever wants to raise their hand and do that. That'll be at the end. And then at the end of that, you'll be free to go meet us all at the poster session. So here we go with the 2022 IEP Annual Workshop lightning talk session. And our first speaker is Miles Daniels from the National Oceanographic and Atmospheric Administration. Miles?
Great. Can you see my slides?
Yes.
Great. Well, hello, everybody. Thanks for joining today. So I'm going to be talking about a project that's not done, but just started for the most part, called Monitoring and Modeling Pathogen Exposure in Salmon Migrating to the Delta. This work was funded by California Department of Fish and Wildlife Proposition 1, as well as some assistance from Metropolitan. And this is just a partial list of the people involved. So this is a really large collaborative project that we're doing that's been really instrumental to answer some of the questions we're looking for. So off to the right is the domain of interest for this project. This is showing you the Sacramento and the Feather Rivers. And as you guessed by the title, we're looking at pathogens. So the big picture of this project is really to get a better understanding of how pathogen exposure to other environmental stressors affects the outmigration success of Chinook salmon in the Central Valley. And we're doing this kind of in a two-pronged approach. One is you're trying to collect monitoring data on pathogens and disease in the system, and then also use that data to build models to represent how we think pathogen exposure is affecting outmigration success. And that kind of is represented here on this figure here, known as the epidemiological triad. So whenever you're dealing with the diseases, you can kind of conceptualize your model ideas in this kind of figure where we have a host, we have a pathogen and the environment, and they're all kind of interacting with each other to ultimately have an outcome in the host. So the host for this project is outmigrating Central Valley salmon. The pathogens that we're interested in are a few. Primarily, we're looking at Serotinova shasta, or C. shasta, as it's a pathogen of known concern in the system, but we're also looking at other parasites, bacteria, and viruses. And then the environmental conditions we think are kind of mediating those host pathogen interactions are things like water flow, temperature, duration of exposure, et cetera. And so we're trying to kind of build a conceptual model and a physical model, simulation model of all these factors and how they affect salmon. So how are we doing this? We've kind of set it out in four main objectives, which are shown here. First thing is we want to screen the environment to understand what kind of pathogens and what levels they are in the environment. We're screening these 12 sites shown here off to the right. We go there about every couple of weeks, collect water samples and test those water samples. And then at a subset of these sites, we actually do what's called sentinel fish studies, where we take hatchery-raised fish, put them out in the river, expose them to ambient river conditions, and screen their tissues for pathogens and disease outcomes to get a sense of what's happening. That's only part of it, though, because we want to understand what's happening to wild fish as they move downstream, not just fish sitting in a cage by the river. And so we also work with folks like at the U.S. Fish and Wildlife Service up in Red Bluff to screen some of the wild fish they capture in their rotary screw traps. Then we can take that information, that kind of empirical data about what diseases and pathogens are in the system, and use that to inform a model that we're developing to simulate disease transmission in the system and health outcomes. And that's part three. And then part four is to take all of this, hopefully build it into a decision support tool, kind of a web-based decision support tool to not only share the results, but also kind of share the model and allow people to perturb inputs as they want and see what the effects might be. So real quickly, I'll go into the disease transmission model just a little bit, if that piques your interest. So this is a conceptualization of it off to the right. Essentially what we're doing is we're taking a movement model that's really based off acoustic telemetry data of fish and understanding how they move downstream at different times of the year, at different rates, et cetera. And then merging that with an exposure model, what we call an SIR compartment model, where we break out the population into an S susceptible, I infected, or R recovered or removed fish from the population. And depending on how they're exposed to certain levels of pathogens, other stressors like water temperature, et cetera, that determines how they transition to these different states. For this project, we're really just focused on C. shasta, but this framework can be applied to other pathogens as well. And so ultimately, the goal is to kind of just get better insights into how diseases, pathogens are impacting populations, which are sometimes hard to observe in the field. And that's all I have. And if you have questions, feel free to reach out to me. And thank you very much for listening.
Thank you, Miles. Our next speaker is Allison Brady from the California Department of Water Resources.
Welcome, and thank you for attending this update from the Yolo Bypass Fish Monitoring Program. Just a few months ago, our program had the exciting opportunity to confirm the presence of a species newly introduced to our survey area. This newly confirmed species of killifish is visually similar to one already found in the bypass, and so this may not have been possible without modern genetic technology. Both species of killifish now found in the Yolo bypass are native to the eastern United States, and the long-term effects of their establishment on the Bay Delta system has yet to be fully understood. So, a little background. The Yolo bypass maintains regular beach seine sites as well as a rotary screw trap along the Tule or Tow Drain Canal. Here we have observed rainwater killifish, or Leucaena parva, since 2011. But our killifish catch has increased dramatically since 2020. We started to wonder, what is going on with these killifish? Around the same time, a new paper from Maharja et al. Was published in 2020 that stated a new species of killifish had been recorded at the Delta Cross Channel as early as 2017. We decided to genetically test for the presence of this new bluefin killifish, or Leucaena goodii. What we found was that we most certainly do have bluefin killifish in the system, and that they made up a significant percentage of what we were already recording as rainwater killifish. Around 3 out of every 10 samples came back with confirmation of bluefin DNA. That being said, our current methods test mitochondrial DNA, which is inherited solely from the mother, and so cannot accurately assess the possibility of hybridization or crossbreeding at this time. That possibility of hybridization is particularly interesting to our program, as the visual identifiers used to differentiate the two species appear to be less pronounced in our population. In theory, the dark horizontal stripe spanning from head to tail should be an identifier of the bluefin killifish species. But this has proven to be an unreliable distinction in our field sampling, as is shown with these samples, which have come back with the genetic results that contradict our initial field hypotheses. In the light of this new information, we have adapted our program. We have started recording all killifish species to genus while in the field and began regularly subsampling killifish at our sites for genetic analyses. We hope to continue using this exciting technology to better inform the adaptive management of our program in the future. I'd like to give a big thank you to the UC Davis Genomic Variation Lab for running all of our DNA samples, the Department of Water Resources Aquatic Ecology Unit for running the Yolo Bypass Fish Monitoring Program, and specifically Emily Hubbard for providing data for the figures used in this presentation, as well as Brian Maharja, a former member of our program, for bringing this newly localized species to light. And of course, the Interagency Ecological Program for hosting this event and being a great audience. My name is Allison Brady, and you can reach me at my state email at water.ca.gov. Thank you.
Thank you, Allison. Our next speaker will be Julie Thayer from the Farallon Institute.
Hi, I'd like to talk today about implementing ecosystem considerations in fisheries, specifically contributions of IEP data. We've been using IEP San Francisco Bay midwater and otter trawl data for multiple projects, including trophic interaction studies of Alcatraz seabirds, and 2 projects I'll summarize today: management of Pacific herring and California halibut fisheries. Fisheries independent indices are crucial for management, as fishery-derived indices are subject to many biases. This is particularly important to realize the Marine Life Management Act stipulations of including ecosystem considerations in California fisheries management. The first example I'll give is of Pacific herring management for the San Francisco Bay fishery. Herring are coastal species but spawn in bays and estuaries. We developed stoplight recommendations from ecosystem considerations on the left to guide adjustments to the quota set from the harvest control rule on the right. The herring ecosystem matrix is built of two main components. First, the predicted spawning stock biomass on the left is used to determine the quota on the black line on the graph on the right, and then the ecosystem indicator can potentially adjust the quota up into the green region or down into the red. Yellow suggests caution, but not necessarily a change to the quota unless other warning signs are observed. Age 0 herring catch from the IEP monthly midwater trawls supplies recruit information that allows for biomass prediction, supporting the base of the whole matrix. Specifically, this age 0 abundance from trawls 3 years prior is combined with San Francisco Bay herring spawning stock biomass, or SSB, from the previous year, and also local to basin scale integrated multivariate ocean climate index, or MOSI, which includes sea surface temperature, among other variables. The resulting model is highly significant with good predictive power, and you can see in the graph here the observed, or black line, versus predicted values in gray. My second example is using IEP data for management of the northern portion of the California halibut stock. California halibut are also a coastal species, yet juveniles rear in bays and estuaries. We found intermittent pulses of juvenile abundance, which were related to warm water events. In our modeling, the best explanatory variable, the multivariate ocean climate index, or MOSI, integrates SST with other regional environmental variables, but you can see from the second and third models here in the table that SST is a large component of the explanatory power. This is really important as temperature influences key physiological processes such as growth and maturation, conferring a survival advantage. We're currently working on developing this for predicting recruitment pulses after warm water events. So in summary, ideal indicator components are simple and low cost. They utilize existing monitoring data and syntheses, and they're geared toward a specific management cycle, be it annual or seasonal, etc. IEP data are important for management of fisheries, both in and outside of San Francisco Bay. And so sufficient resources for continued sampling and appropriate backup vessels to avoid data gaps cannot be overstated. Herring predictions were compromised due to late 2010 data gaps, and understanding was also compromised of the longer than normal juvenile halibut pulses that we observed during the blob marine heat wave. And this can have really important ramifications for understanding effects of climate warming on this important fishery. So in conclusion, I'd like to thank the California Department of Fish and Wildlife and the Pew Charitable Trusts for supporting this work. And I can be reached at the email address here. Thanks very much.
Thank you. Our next speaker will be Dulcinea Avouris at the University of California, Merced.
Hi, everyone. Sorry about that. I lost my unmute button. I'm a postdoc at UC Merced working with Aaron Hester, and I'd like to thank IEP for this opportunity to share the Mercury from Space project, which is funded by the California Department of Fish and Wildlife with Prop 1 funds. This project is also a collaboration between UC Merced and the USGS, and the primary people at both the California Water Science Center and Menlo Park are listed here. And in addition, quite a few people from the Water Science Center participated in our field campaigns, and I'd like to acknowledge them as well. So the goal of this project is to develop cost-effective monitoring technologies to better understand the production, fate, and transport of mercury in the San Francisco Bay Delta. As I'm sure most of you know, mercury is an ongoing environmental hazard in the Delta, and that there are multiple state initiatives to reduce methylmercury exposure in fish, wildlife, and human populations in the area. Most fish consumers in the Delta may be taking in more than the US EPA maximum dose. And we also know that methylmercury has a large bioaccumulation factor, and that in addition, concentrations of methylmercury in fish are highly correlated to aqueous methylmercury. So how do we use remote sensing specifically to help meet this challenge? So remote sensing provides free landscape scale measurements of in-water constituents every few days. It's been shown that mercury and methylmercury are correlated with turbidity and CDOM in the Delta. And we can measure both of these water quality parameters from satellite-based sensors. So the remote sensing measurements of both turbidity and CDOM can be leveraged then to model the relationship between mercury species and sediment or CDOM. These models, in turn, provide insight into better understanding mercury cycling in the Delta and allow us to visualize the distribution and concentration of mercury without costly field sampling. In order to build these models, however, we need a sensor network of both in situ measurements and current remote sensing data. So this project has collected an extensive data set of water quality metrics, including, of course, turbidity and CDOM, but also measurements of other parameters such as salinity, stage height, temperature, etc. The data sets that we've collected are both temporally and spatially dense, and these field campaigns stretched from mid-2019 through spring of 2021. So this project, the conceptual design for this project here is on the right, showing the sensor network that we leveraged and the kind of relationship that we're expecting between both sediment and CDOM, or dissolved organic carbon, and mercury species. On the pictures of the USGS teams deploying the various sensors that were used during our field campaigns. In order to build, so because we know that particulate mercury species are correlated with turbidity in the Delta, it was important to make sure that the remote sensing measurements of turbidity were correct. So this figure shows a Sentinel-2 image from July 8th, 2020, which was one of our field campaign dates. The standard algorithm that's used to calculate turbidity from remote sensing measurements that we initially used yielded really good results with an R-squared of about 0.68 when we validated it against USGS continuous monitoring station turbidity measurements that were spatially distributed through the entire Delta. However, we were able to retrievals or the turbidity calculations when we tuned that turbidity algorithm using measurements of attenuation and absorption that were collected during our field campaigns. The final R-squared for our tuned algorithm is 0.78, which is slightly better than the most recently published remote sensing turbidity validation for the Delta. So this turbidity measurement then becomes one of the input variables that we use in our models. Once that turbidity algorithm was refined, then we started developing these models between remote sensing data and in situ mercury measurements. And we started these models only looking at the information that we can obtain from remote sensing imagery. So here are two of the initial models for particulate total mercury. We, of course, expect this species to be correlated with turbidity. But as you can see, these models are not very good yet. The random forest model, which is on the left, explains about 25% of the particulate mercury. And the multiple linear regression, which is on the right, explains only about 21% of the mercury. However, as initial models, these are very promising, and we're working on refining them by factoring environmental variables such as season and salinity, as well as incorporating more in situ measurements. So thank you for this opportunity, and that's all that I have.
Thank you. Our next speaker will be Sienna White from the San Francisco Estuary Institute.
Perfect. Okay. Does this work? Great. Okay. Hi, everyone. My name is Sienna White. I'm an environmental analyst at the San Francisco Estuary Institute, and I will be telling you a little bit about some of the work we've been doing modeling hydrodynamics as well as biogeochemistry in the Delta. This is my team here. I've been working with Pradeep Maganthan, Ali King, Farid Karimpour, as well as Dave Sen. So a little bit of background, which I'm sure you all know. The Delta is obviously a really important system for both ecological as human health. It's really highly altered, it's complex, and phytoplankton in this region is really hard to model because there's kind of this ongoing debate about whether or not it's controlled purely through light limitation or through some combination with heavy benthic grazing. So it's a really interesting area to study. We've been working on developing a couple of the numerical models so we can kind of get at some interesting science and management questions from a really quantitative lens. And yeah, this work has kind of been going on for about the past 5 years or so. So in terms of the model that we're using a coupled model. So this means it's a hydrodynamic model coupled to a biogeochemical model. And the modeling suite you use is DFlow-FM for the hydrodynamics and then DELWalk for the biogeochemistry. These are both from Deltaris in the Netherlands. And our domain for our model sort of spans all the way north to the Sacramento region, south near Stockton, as well as all of San Francisco Bay. And it takes forever to run. We've got about 75,000 cells in the horizontal, 10 vertical layers. And on wall clock time for a full water year with 2 months of spin up, it's about like 5 to 6 days on a high powered computer. And as of now, we have 2 water years that we've been looking at with this model. So both water year 2011 as well as water year 2016. And when it comes to the processes we're looking at with this biogeochemical model, it's sort of the whole suite of processes you'd expect. We've got nutrient cycling, we've got sediment diagenesis, we have phytoplankton growth and respiration and mortality. And I want to point out that we are modeling grazing in this model, both pelagic grazing from zooplankton, as well as 2 species of bivalves, both Patema corbula and corbicula clams in our model. And so I got kind of ambitious with the lightning talk. I will show you a couple of validation plots. So this is some validation with discrete monthly data from EMP and USGS. So from all these sites here, and we can use time series. So looking at the comparison between our model and measured data for validation. And so to give you an idea of right now where we're at for dissolved inorganic nitrogen, so summing up the nitrate and ammonium, we have 3 stations here in Sassoon Bay, D6, D7, D8. D19, D26 near Frank's Tract, and then 657 is on the lower Sacramento. And what you can see here is we put both water years side by side because in trying to calibrate our model, we're interested in developing a global calibration. So trying to represent processes with the same set of parameters across 2 very different water years to feel confident that we're kind of nailing down what this process is doing in the Delta. And this purple dots, these are our measured data from EMP or USGS. And then this dark blue line is our modeled results at that same point, down to a daily average, and the shading behind it is a higher frequency signal. So you can see here in a really strongly tidal system, you're going to get a lot more daily range. And what you can see here is across 2011, which was really wet, and 2016, which was on the end of the drought, our model is able to replicate both of these signals pretty well, especially when we think about seasonal time scales, the general magnitude of this peak as well as the timing of it, and then this drawdown in the spring. We're excited about this validation. We're seeing a pretty good capturing of that. Chlorophyll is a slightly different story with our model here. So you can see in 2011, there was some pretty major bloom activity in the whole region. And in 2016, there's also bloom activity, but it was a little more short and kind of spikier. And in 2011, our model is able to replicate the general sort of timing and magnitude of some of this higher biomass. And then in 2016, we're missing some of these isolated events. So kind of ongoing work is thinking about, what factors led to these kind of spiky bloom events in 2016 and how can we capture them? But kind of generally speaking, I guess, the thing that's exciting is now we're able to develop this model. We can now start to think about how we can apply it. So thinking about management questions, we actually did a study in the last year thinking about how the regional sand upgrade will affect nitrate and ammonium loading and concentrations in the Delta region. And we can also start to think about science questions. And so one of the projects we're going to be working on in the coming year is thinking about what are the drivers of primary production in like Sassoon Bay, and using our model to kind of think about that balance between, let's say light limitation compared to benthic or pelagic grazing. And so kind of to summarize is, we've been working on this model. We have validation, and we feel confident that especially on seasonal timescales, we're able to replicate DIN and general kind of chlorophyll, especially ambient chlorophyll without bloom activity in the region. And we're excited to kind of apply this model and continue to work with a lot of our collaborators. And I want to acknowledge some of them here. We've gotten a lot of great technical input as well as funding. So with that, I will close. Thank you so much.
Thank you. Our next speaker will be NC Habibi from the University of California at Davis.
Hang on 1 minute, Steve. Encountering a technical difficulty.
All right, insert game show music here.
Yeah, for some reason I'm getting an error message. NC, are you able to do your presentation live?
Wait, was that a question for me?
This is for NC.
Oh,
We're having a
sure.
little trouble with her presentation.
NC, are you able to pull up your presentation and do it live?
Yeah, I'm trying to figure out how to share it.
Oh, okay. Use the green share button at the bottom of the screen.
Yeah, but it's not showing my presentation. I don't know why.
Do you already have it open up on your screen?
Yes, it is ready, but
And you're clicked on the basic tab up. There's usually 3 tabs, basic, advanced files. Are you in the basic tab?
yes, I am.
Okay, you should be able to, if you have your PowerPoint already open, you should be able to see your PowerPoint presentation.
It is not showing the PowerPoint presentation when I push the share button. It's
John, do you have any advice?
you're not seeing it, right?
Well, we're seeing your screen right now. So we're seeing, there it is.
Oh, okay. Sorry about that.
No, we're good.
Hello, my name is NC Habibi from University of California. I'm presenting a part of my PhD project, which is domestication selection happening in the refuge population of Delta smelt? Delta smelt are managed at UC Davis Fish Conservation and Laboratory FCCL since 2008, 1 year before they were listed as endangered in California. And FCCL is a conservation hatchery, and goals of conservation hatchery is to maintain the refuge population as close as possible to wild population to decrease the demographic and genetic consequences of captive propagation. For this, they are in collaboration with Genomic Variation Laboratory at UC Davis for parentage analysis and pedigree construction, where they use genetic markers, microsatellite markers, to assign offspring to their parents and then pair male and female for each cross. Despite the intense genetic management at the FCCL, there are evidence of domestication selection. In a study by Finger et al. Showed that crosses that have 2 captive parents, showing in pink here, they have higher fitness in compared to crosses that have 1 captive or no captive parents. And as we go through generations in captivity, this fitness is increasing. So the questions are, can we detect heritable genetic changes as a result of the numbers of generations in captivity at the FCCL? And if we can, when is it happening, in the early, medium, or late generations in captivity? To answer those questions, we collected 240 FCCL archive samples, and we divided them into 4 groups based on their DI value. What is DI value? DI is a domestication index, which is the measure of each individual's hatchery ancestry. And it's calculated by taking the average of parents' DI plus 1. Why 1? Because the fish itself spent 1 year in captivity. For example, if we have a culture fish with 2 white parents with DI of 1, and we cross it with a white fish, the offspring from that cross will have a DI of 1.5, which is the average of parents' DI plus 1. And as I said, we divided them into 4 groups as low DI, medium DI, high DI, and wide group, which has a DI of 0. And we use restriction site associated DNA sequencing, RAD sequencing, to collect genomic information from individuals. And then we align it to Delta SMART reference genome that's recently created. And we collected more than 100,000, we discovered more than 100,000 single nucleotide polymorphism. And we use this for our genetic analysis. To find the regions that are under selection across genome, we use FSD, which is a measure of genetic differentiation among groups. And based on our study questions, we did 2 comparisons for our FSD. In comparison 1, we compared the wide and low DI group with medium and high DI group to detect loci that are gradually changing in captivity. And in comparison 2, we compare wild group with our hatchery group, which include all the DI group in the hatchery, to detect loci that are rapidly changing in hatchery. And as a result, in comparison 1, we found 2 regions that had a significant FSD on chromosome 15 and chromosome 22. And surprisingly, in comparison 2, on those 2 regions showed up with a higher difference in FSD in compared to other peaks across the genome for that comparison. Since they appear in this comparison between wild and hatchery group, that means domestication selection is happening in early generations. Right now we are, and after we had 2 candidate regions, the 15 and 22, since they were appearing 2 comparisons, they were candidate region for domestication selection, and then we examined them for error possibility and sequencing artifact, and as a result, the region on chromosome 22 was qualified as a region under selection for domestication. Right now, we are exploring the region to find specifically which gene or genes are under domestication selection, and hopefully by finding the genes and biological pathway and that are involved in the process of adaptation in captivity, we can help FCCL to modify environmental conditions that are causing the domestication. By that, I will thank all the people from our lab and involved in this project, especially the state water contractor for funding this project, and IEP conference to giving me the opportunity of presenting. And thank you so much for listening. And stop share.
All right, thank you.
All right.
Our next speaker will be Mallory Bedwell from the California Department of Water Resources.
Hi all, thanks for coming and welcome to the jungle. My name is Mallory Bedwell and I'm going to take you on a somewhat musical journey examining the preliminary results of the 2021 North Delta Food Subsidy Monitoring. So the purpose of the North Delta Food Subsidy Action is to use existing infrastructure in order to redirect water through the Yolo bypass and to increase net positive flow during the fall and summer months. And to move quality food downstream to smelt habitat in the Cache Slough complex. The North Delta Food Subsidy Action is an adaptively managed project that takes into account water year type, water source, in order to move water through the ecosystem. And it's important for managers to know when to conduct actions, and these dry years with no actions can help us to understand baseline conditions And our question for this past year was, should we conduct actions in dry years? So for 2021, this was a dry non-action year, which allowed for us to conduct baseline monitoring, like I said, to use as a comparison to help inform our adaptive management plan. And so we conducted biweekly sampling at 10 sites total during the summer fall period when a flow action would typically be conducted. And so we looked at flow, took both discrete and continuous water quality measurements, and took both phytoplankton samples as well as 150 micron zooplankton tows in order to examine lower trophic response. We also monitored non-target species and interacting biotic effects like weeds. So we're just getting some of these results back. So this presentation will just focus on continuous results. For 2021, we did observe high amounts of aquatic vegetation that were not only problematic for taking data, which you can see in these photos at left, but may impact things such as available light, flow, nutrients, and could lead to changes in biota like plankton. So let's look at the measured flow through the system. On these plots, the pulse period is highlighted in gray. So in 2021, we did not have a managed flow pulse, but we did have a small natural pulse due to local ag drainage in the bypass. And so you can see it lasted for about 4 days here. We experienced some very dry conditions and had extremely low flow throughout much of the action. What you can see it right here, this is Rominger Bridge, which is our uppermost site. And we previously had not known that there were submerged abandoned cars in the canal that we had been towing over for the past couple of years. So that was really interesting to learn. And then when compared to 2019, you can see that there was this large sustained flow pulse, and that was due to it being a wet year with a managed flow action. These are plots of DO concentration during 2021 and 2019. The black line indicates DO threshold for salmon health that we are trying to avoid not only for salmon, but for other native species in the bypass. As you can see, sites above Lisbon Weir consistently had low DO even before the action would even take place. And this green line here is I-80, and you can notice that there's very low DO throughout the whole monitoring period. Compared to 2019, we do see a DO during the action, but there are better conditions overall due to it being a wetter year, and you can see more of those lines above the threshold. We also do see a DO increase later in the season after the flow pulse, around the time when we would expect fall run salmon to enter the system. So based on what we saw in 2021, we've identified considerations for conducting actions in dry year to help with our adaptive management plan. And so we want to take into account water that's available in order to create the flow pulse. And, this might be more difficult in dry years. We also want to look at initial DO in the system because there is concern about low initial values. And we want to see if there are large amounts of aquatic veg, which could potentially reduce the benefits of the action. And with that, we would just want to say thank you to all our collaborators and everyone that has helped out with this project. Thank you everyone for your attention and enjoy the rest of the conference.
Thank you. Our next speaker is Jason Hasrick from ICF International.
With state and federal endangered species protections in place, why aren't winter run recovering? Less than 5% of winter run in most years make it to the ocean, and that's less than most of the other Central Valley runs. The purpose of this study is to unpack why and where winter run mortality tends to be higher. By modifying an existing acoustic telemetry array to cover the upper extent of anadromy to the delta over the winter outmigration period, we conducted a MARC recapture study on hundreds of fish every year over a period of 5 years, starting in 2013, which happened to coincide with a prolonged drought that culminated in one of the wettest years on record. Using AIC model selection, we evaluated a suite of models that included combinations of fixed habitat covariates, such as the number of diversions, tributaries, the percent of wooded bank habitat type, and the extent of off-channel habitat. We also included dynamic covariates, such as the mean annual flow and NOAA's raft model outputs for reach flow, reach velocity, and reach temperature. For the fixed habitat covariates, we observed that some features were more positively associated with survival in the upper part of the river, and then switched to being more negatively associated with survival in the lower part of the river, or vice versa, whereas they were more positively associated in the lower part of the river and more negatively associated in the upper part of the river. This suggests that these fish were switching behaviors and interacting with these habitat features in different ways, such that they're rearing in the upper part of the river and migrating in the lower part of the river. Another thing I wanted to draw your attention to is that survival tends to dip in this lower part of the river, starting at Red Bluff and then going down to Tisdale. This also coincides with a stretch of the river that happens to have a lot of bare bank associated with it. And this bare bank turns into off-channel habitat in 2017. So when flows are high, fish tend to stay in the river. In 2017, we found that fish stayed in the river longer than in any of the other years. When flows are really low, they will tend to leave on these pulses, except when there's no pulses at all, and then they can stay at very low numbers throughout the system. But they don't survive very well in 2013. In 2013, we saw the lowest survival compared to all the other years. The other thing I wanted to show you here is that if we look at the beta estimates from the model, from the top ranked model in the model selection, we see the annual flow was most positively associated with survival. And this width to depth ratio term was most negatively associated with survival. These variables in the middle did not have any kind of conclusive effect on survival. So what I'd like to talk about now is why it is that we see a strong positive effect of annual flow and a negative effect of this interaction term between annual flow and intra-annual reach flow. So as you might expect, survival tends to increase with annual flow to an asymptote where increasing flow does not have any measurable effect on survival. But if you look at this another way and you look at the slope for this intra-annual reach flow against the mean annual flow, for each of these years in the study, 2017 is shown here in light green, and these years during the drought are shown here in these darker colors. And you see that there's this strong effect where in really low annual flow years, there's a strongly positive benefit to having higher reach flows or pulse flows, so that predicted survival as a function of reach specific flow is much higher in these low flow years than we might see in a high flow year. And so managed pulse flows might be a very good way to improve survival during drought years, such as the one we're in now. Thank you very much.
Thank you. Our next speaker is Katarina Pien from the Department of Water Resources.
Hi, my name is Katarina Pien from the Department of Water Resources, and today I'll be talking about some of the results from our continuous water temperature synthesis. So my questions for this talk are, what are spatial and temporal trends in delta water temperature in the last decade? And how often do delta temperatures exceed heat stress thresholds for species of interest? Looking at general trends, we ran models for mean, max, min, and range temperatures. And you'll see that it is generally warmer in the central and south delta. So another reason this region is not suitable for delta smelt or for restoration. Generally cooler in the confluence north delta area, which is an area that is experiencing a lot of restoration. So good to know that temperatures are a little more suitable where this is occurring. And in the summer, Sassoon Bay also seems to provide slightly cooler habitat as well. And then range is greater in the summer months, spring, summer months, and in Sassoon Marsh. So to tie these trends to species of interest, we can look at temperature thresholds of different species. In the top plot here, you can see in the larger font, the non-natives falling out at higher temperature thresholds, around 30 to 40 degrees, and the native species falling in this bottom area between 20 and 30 degrees Celsius. So many non-natives have higher tolerances compared with non-natives. Meanwhile, if we're looking at suboptimum temperatures, which is a little more relevant to what might be experienced in the wild, a lot of these tolerance studies might be done in the lab. So these are temperatures at which the organisms might be feeling stress, so that maybe they're not experiencing mortality, but stress can affect their spawning habits, their growth and metabolism, forcing them to eat more. So we really care about these suboptimum temperatures as well. You see that for many natives, they're starting to experience this kind of stress around 20 to 21 degrees Celsius, whereas some of the more heat-tolerant natives and non-natives experience this closer to 25 degrees Celsius. And when you see some of these non-native vegetation, clams, this is when they're starting to experience habitat that is suitable for them. So really above that temperature is a good temperature for them. So to figure out how many days of stress these species are actually experiencing, we ran some GAN models for 21 degrees and 25 degrees thresholds of stress. And our definition of stress is that there are at least three consecutive days reaching these thresholds in a row. So for these species, in an average year, you're seeing 60 to 96 days of stress. In an average year, 89 to 105 days of stress in a warm year, with a difference of about 34 days in these Suisun Bay and South regions. And you'll notice that the entire delta is actually experiencing at least 2 months of stress in an average year. All of this occurs between April and October, so in these warmer months. The whole delta is stressful. So at the 25 degrees threshold, you do see a lot less of this stress occurring in the northern part of the delta, but still about 44 days in the south and central delta, 66 days in a warm year in those regions, with a difference of about 30 days. So some takeaways here. The whole delta is experiencing water temperatures, at least in certain months, close to native species thresholds, especially in the south and central delta. An increase in these warm and drought years, which we saw in the example compared to the normal year, is likely to push species and water management to their limits. These warmer years are going to bring more suitability for harmful algal blooms, aquatic vegetation, and non-native species in warm months and regions, and may further alter habitat and suitability for native species. There does seem to be potential temperature refuge, cooler temperatures in the North Delta, Suisun Bay, and Marsh, where restoration is occurring, but all regions are close to their thresholds in at least part of the year and will likely need water management to remain below temperature thresholds. Thank you so much, and please let me know if you have questions.
Thank you. Our next speaker is Calvin Lee from ICF International.
Hi, my name is Calvin Lee. I'm a senior biologist at ICF, and I work on the Directed Outflow Project for the U.S. Bureau of Reclamation. Today, I'm going to be talking to you about our research looking at zooplankton communities in the Sacramento Deepwater Ship Channel and how they differ spatially. The ship channel is considered a hotspot for delta smelts. Previous research has also shown there are different environmental properties in different portions of the ship channel. We asked if there were different zooplankton communities in the different parts of the ship channel and what variables were driving these differences. In our study, we used data collected during the fall season from 2017 to 2020. The map on the right shows sampling locations for those 4 years. In the field, we collected zooplankton, phytoplankton, and water quality data. Today, I will only be going over water quality and zooplankton data. We divided the ship channel into 5 different regions based on both turbidity and specific conductivity. The map on the right highlights the different regions. The figure on the left shows that turbidity, represented by the solid black line, increased as we sampled upstream and peaked around channel marker 66 before decreasing further up in the ship channel. Specific conductivity, represented by the dashed line, increased as we sampled upstream. We interpreted conductivity as a proxy for water residence time. The longer water sits in a region, the more it evaporates, which explains the higher conductivity seen in upper portions of the ship channel. In the rest of the figures, we will be using the same color scheme to represent the different regions of the ship channel. These figures show the average values for different water quality variables by region. Within each plot, regions that share the same letter are statistically similar, while regions with different letters are significantly different. We saw that chlorophyll A and phosphate was higher in the upper regions, while nitrate and ammonium were lower, moving upstream. Here we have an NMDS plot. Each polygon represents a different region of the ship channel. Polygons that overlap indicate zooplankton communities that are similar. The figure shows upper and lower portions are different, with the middle region acting as an intermediary. The variables that drove these differences were specific conductivity, again a proxy for water residence time, chlorophyll A, phosphate, nitrate, and ammonium. The figure on the left shows that the upper regions of the ship channel had higher average biomass. The figure on the right shows that the proportion of certain zooplankton taxa, such as Pseudodiaptomus forbici, decreased moving upstream, while other species, such as Siennaw dorii and various other Cladocera, increased. We found downstream regions differed from upstream regions of the ship channel. Specific conductivity, chlorophyll A, and phosphate increased as we sampled upstream, while ammonium and nitrate decreased. Our NMDS analysis showed that conductivity, a proxy for water residence time, was an important factor in determining where zooplankton communities were found. We also found higher zooplankton biomass in the upstream regions where the community was dominated by Cladocerans and Cynocalanus. We conclude that there is a higher biomass and a different zooplankton prey community available for delta smelt in the upper portions of the ship channel. However, previous diet studies of delta smelt found that Cynocalanus and Cladocerans were more commonly found in the gut during winter and spring. I'd like to thank my co-authors April Smith and Andrew Kalmbach, as well as ICF field staff and biologists, the U.S. Bureau of Reclamation, state water contractors, U.S. Fish and Wildlife Service, Randy Dahlgren, Steve Sidaro, and Zine Wang at UC Davis for helping process our nutrient samples. Thank you.
Thank you. Our next speaker will be Becky Stanton from the Office of Environmental Health Hazard Assessment.
Great. Good afternoon. I appreciate this opportunity to talk. And sorry, it looks like you're seeing, is that the correct presentation? This opportunity to talk about the Interstate Technology and Regulatory Council's harmful cyanobacteria bloom teams that I've been an active member on, and also put a shout out for the session Jenna organized on harmful algal blooms happening on Thursday, and look forward to participating on that as well. So our HCB team with ITRC has a website currently. I'm here representing that team, as well as my co-team leader, Ben Holcomb from Utah, and our program advisor, Sherry Basinger. And so our existing guidance that is out currently covers cyanobacteria in general with a greater focus on planktonic cyanobacteria, and covers an overall introduction to cyanobacteria, monitoring methods with a selection tool, methods for communication response, a management criteria tool focused on in-lake management, a focus on nutrient management, which what I know has come up a lot today, some recommendations, and a visual guide. And we are doing training on that starting again April 26th with a link that I can put in the chat later, and that's 10 a.m. Pacific. What's also out now is a harmful cyanobacteria identification video that was put together by several colleagues, walks through how to identify cyanobacteria based on what you see coming to a water body, as well as some simple field tests, and also talks about what other things you might see in the field that are not cyanobacteria blooms and how to separate those out.
Hey, Becky,
Yes.
we're still on your first slide.
Okay, it shows like it's moving there. Okay.
There it is. Now it's moving.
Okay.
We're on the HCB identification video slide now.
My apologies. Thanks for letting me know. And so benthic cyanobacteria are unique from water column or planktonic HCBs, as they can occur in clean water and with higher flow. They may be less visible from the surface and can co-occur with green algae. Conditions such as flow and temperature or aquatic weeds, as was talked about earlier, can affect the growth differently. And they've certainly been associated with both human and animal impacts, particularly dog deaths. And we more typically see neurotoxins such as anatoxin A. And so the benthic HCB guidance document is actually expected out very soon. It covers the unique ecology of benthic cyanobacteria, a more in-depth discussion of cyanotoxins that'll be shared with both documents. We have information specific on how to monitor for benthic cyanobacteria and have updated the visual guide to include those taxa. We've updated our management strategy sheets to incorporate benthic cyanobacteria and additional water body types, and focus on the unique aspects of messaging about benthic harmful cyanobacteria blooms. And that'll be the first round of our live training on that document. That'll be April 28th at 10 a.m. And we welcome people to register for both trainings. And that is it. Thank you.
Thank you, Becky. Our next speaker is Nina Pack from the University of California at Berkeley.
Hi everyone, I'm Dr. Nina Pak, and today I'll be sharing a study on Pacific herring, a forage fish species that is commercially fished in the San Francisco Bay. For some background information, in California, the Pacific herring commercial fishery has landed more than 11,000 metric tons since 1980. The largest spawning aggregation occurs in the San Francisco Estuary, where adults move in the estuary from the ocean to spawn on substrates such as eelgrass beds, rocky shorelines, and other surfaces. However, recent evidence suggests that the spawning stock biomass, also known as SSB, is not only highly variable, but also has declined to record low estimates, with changes in the environment such as salinity and temperature fluctuations implicated as potential drivers. In this study, we investigated the extent to which salinity and temperature fluctuations and trends, demographic effects, and density dependence affect Pacific herring population dynamics by looking at long-term data sets from CDFW San Francisco Bay Study and egg surveys from 1981 to 2015. Specifically, we asked, how do salinity and temperature fluctuate over time? How do Pacific herring age classes fluctuate over time and among regions? How do they respond to salinity and temperature fluctuations in each of these regions? Are there spawning effects, recruitment effects, or a combination of both? And lastly, is there evidence of density dependence? When we averaged monthly salinity and temperature measurements by region, we found that annual fluctuations in salinity and temperature were similar, but the ranges in maximum and minimum values differed, particularly for salinity. In the top right figure, the blue bars indicate when average conditions are cooler or fresher, and the orange bars indicate warmer or saltier conditions. As we expected, we see that South Bay and Central Bay were saltier, while San Pablo and Sassoon Bay, which are further inland, were fresher. We ran multivariate autoregressive state space models, which are time series models that use the Kalman filter. When we compare age class trends, we see that age 0 fish were common in all 4 regions, South Bay, Central Bay, San Pablo Bay, and Sassoon Bay. We found that most age class abundances lacked trends in variability and showed boom and bust cycles, with the exception of age 0 fish in Central Bay and San Pablo Bay, which showed evidence of population growth over time, surprising us all. From these models, we observed a positive association between age 0 fish abundances and cooler and saltier conditions in certain regions, and positive associations between age 1 fish abundance and saltier conditions in Central Bay. In contrast, we see no association between SSB and salinity and temperature. In addition, these models showed no support for recruitment or spawning effects, but showed strong support for density dependence in age 0 and age 1 fish. Overall, abiotic drivers, including temperature and salinity conditions, as well as density dependence, largely explain the observed abundance fluctuations of juvenile Pacific herring. Our results match the larger narrative of Pacific herring being a coastal marine fish. We see that age 0 fish were positively associated with cooler, saltier conditions. Age 1 fish were also positively associated with saltier conditions in Central Bay, and SSB was not influenced by temperature or salinity. In addition, evidence of density dependence suggests that biological factors such as spawning substrate or food resources were limiting population growth for juvenile life stages. In general, our results indicate that Pacific herring population dynamics were driven by hydroclimatic variability and density dependence. Under climate change, they may benefit from drought conditions. However, they may be vulnerable to marine heat waves that affect core nursery habitats such as Central Bay. We recommend that ecosystem-based fisheries and management focus on improving the carrying capacity of the estuary via habitat restoration, food web recovery, and reducing harvest during poor ocean conditions via fishery closures. Thank you.
Thank you. Our next speaker is Nicole Parker from the University of California at Santa Barbara.
Good day to all IEP attendees. My name is Nicole Parker, and I am a 2020 Bay Delta Fellow and PhD candidate at the University of California, Santa Barbara. Today, I'll share my work to enable the evaluation of contemporary toxic burdens and sources of pesticides within the Bay Delta, as well as risk mitigation opportunities. Pesticide toxicity in California's surface waters remains a widespread and significant concern despite advancements in tools for predicting aquatic health risks. Monitoring campaigns under the Clean Water Act have designated 13% of California's monitored streams as impaired by pesticides, 56% of which are hydrologically connected to the Bay Delta. Available tools for predicting pesticide risk for watersheds near the Bay Delta are limited by either their inability to simulate the cumulative toxicity of the many pesticides in use, to simulate the risk across the hundreds of watersheds and thousands of application sites near the Bay Delta, or to account for the diverse irrigation practices of crops, which is a primary driver of pesticide transport. Another concern is that data is often unavailable for key drivers of pesticide fate within water bodies. To address these concerns, I developed a pesticide exposure assessment framework, the Pesticide Management Prioritization Model, PMPM. Distinguishing features of the tool include predicting pesticides transported to surface waters for watersheds near the Bay Delta, even in watersheds without in-stream hydrologic data, simulation of irrigation method and timing based on irrigation survey data at the farm level, simulation of the cumulative toxic burden of pesticides, auto-integration of pesticide use data for the area and time period of interest from pesticide use reports required for all growers in California, results curated toward the prioritization of opportunities to manage pesticide toxic burdens. Efforts to simulate pesticide toxic burdens in the tool account for the unique crop rotations, climate patterns, irrigation practices, and soil conditions within the Bay Delta watershed. The tool predicts toxicity based on the concentration for each pesticide at which 50% of fish, cladocarans, and benthic invertebrates within a sample experience mortality or immobilization. For the most recent 5-year period of available pesticide use data, I will highlight insights provided by the PMPM for managing pesticides in watersheds near the Bay Delta. Of watersheds near the Delta with agricultural pesticide applications, 79 have increasing toxic burdens. These watersheds were primarily impacted by few pesticides and application sites, which include the active ingredients bifenthrin, clothianidin, and chlorpyrifos. The application sites of greatest impact include wine grapes and almonds. For application sites, the PMPM provides not only an index of the net toxic burden applied, but the mass of pesticides applied, gross economic value, and economic value as weighted by health impacts. Safflower had the worst rating across indices for economic and environmental prosperity. The PMPM also facilitates a consideration of chemical alternatives. For example, consider an almond farmer in the Turlock Lake watershed near the Bay Delta. Using the PMPM, the farmer can observe 80% of the toxic burden is attributable to applications on almonds and 74% from the application of bifenthrin on almonds. Based on these results, the farmer considers their routine use of bifenthrin to combat mites, one of the most damaging pests to almonds in the watersheds. Given this knowledge of the high environmental impact of bifenthrin, they review chemical alternatives. In review of alternatives, they find bifenazate is a pesticide which targets mites and has a substantially lower toxicity profile and environmental persistence. Key benefits of the PMPM to stakeholders of the Bay Delta include the ability to consider grower interest in management decisions, accessibility of information on an app compatible with any device, the tool's ability to efficiently summarize pesticide toxic loads with default parameters and auto-loaded pesticide use reports, and the promotion of data synthesis for improved pesticide management. Thanks for listening. I'd like to acknowledge my research advisor, Dr. Arturo Keller, as well as generous funding provided by the Delta Science Fellowship under the California Sea Grant.
Thank you. Our next presenter is Timothy Musson from Regional SAN.
Hello everyone, this is Tim Musson from the Sacramento Regional County Sanitation District, also known as Regional SAN. Today, I'd like to share with you some findings from a study that Lisa Thompson, James Noss, and I conducted in the spring of 2020, investigating phytoplankton growth in the Delta Cross Channel. Despite elevated nutrient concentrations in the lower Sacramento River, phytoplankton biomass is commonly quite low. Over the last 20 years, the average chlorophyll A concentration measured at Hood by IEP's EMP monitoring program was 2.4 micrograms per liter. The cause for this low chlorophyll A concentration is unknown, but might be due to a combination of short residence time, low light levels, high clam or biomass grazing, or the presence of unknown toxic chemicals. When the twin radial gates are open, water from the Sacramento River enters the Delta Cross Channel near the town of Walnut Grove and continues downstream to the central delta. We monitored phytoplankton growth in the Delta Cross Channel during a springtime gate closure to determine if the increased water residence time might result in higher chlorophyll A concentrations. As shown in the top figure, the initial chlorophyll A concentration in the Delta Cross Channel was 2.5 micrograms per liter, as is typical for the lower river. But after the gates were closed for 3 days, it increased rapidly to greater than 30 micrograms per liter. On the bottom figure, you will see that ammonium and then nitrate concentrations were reduced in the Delta Cross Channel following this gate closure, likely due to the nutrients being taken up by growing phytoplankton. When the Delta Cross Channel gates reopened, chlorophyll A and nutrient concentrations returned to their initial levels. Please note that this study was conducted before Regional SAN implemented our new biological nutrient removal process, which currently nitrifies ammonium and reduces total nitrogen load in our effluent. We also investigated other nearby locations during this Delta Cross Channel gate closure and found that the chlorophyll A concentrations did not change in the Sacramento River mainstem, Georgiana Slough, or downstream of the Delta Cross Channel in the North Fork McCullumee River. Temperature sensors determined that the water in the Delta Cross Channel became thermally stratified during the gate closure. The hashed lines in the figure above represent days when the Delta Cross Channel gates were open and temperature was similar at the river surface and bottom. The solid line furthest to the right shows a 4 degrees Celsius difference in water temperature that occurred on June 11th, when the Delta cross-channel gates were closed. This stratification likely allowed phytoplankton to remain near the surface, where they could grow rapidly in the warmer water and increase light levels, and potentially avoid clam grazing at the river bottom. The phytoplankton biovolume was dominated by diatoms, but more than doubled in taxonomic diversity during the bloom. We thank the Nature Conservancy and the Department of Water Resources for providing levee access to some of our sampling locations. And thank you for your attention.
Thank you. Our next speaker is Christian Denny from the University of California at Davis.
Thanks, Steve. So today I will be talking about validation and application of strontium isotope geochemistry in endangered delta smelt. Our lab uses microchemistry and otoliths, which are ear stones, to track the movements in delta smelt and other species in the San Francisco estuary ecosystem. However, before we can be really confident in how well those chemical signatures are assessing movements, we need to validate the tools used to measure the chemistry, as well as how closely the chemistry matches actual environmental conditions. So in order to test this, we collaborated with the FCCL and used cultured delta smelt and trucked in ocean water to create known salinity conditions in groups of delta smelt. And then after the experiment concluded, we measured the strontium signal in the otoliths. And what we can see here in the top panel is the experimental fish, which were initially reared in 0.1 ppt salinity water, transitioned to 3 ppt salinity water, and then had a second transition to 6 ppt before the conclusion of the experiment, and compared to the control fish, which were reared in 0.1 ppt water throughout the entire duration of the experiment. And so what we can see is that our otolith-based salinities, which you can see here in the bottom left panel, closely match the known salinities of the environment. And secondly, that we were able to estimate the timing of the transition from the 0.1 to the 3 ppt to within about a week to 10 days or so. And so these, while the transition from 3 to 6 was a little more difficult for individual profiles, although you can see in aggregate, there is a change in the strontium chemistry in the mean profiles. It's just difficult to see in individual fish. So taken together, we can say that our geochemical reconstruction of salinity history appears to be relatively robust, especially in low to moderate salinity in delta smelt. So now that we've confirmed the strontium signal in the otolith well matches the conditions that the fish experienced over their life, we can use this tool to look at life histories in delta smelt. And so we did that by expanding on a paper we published in 2019 describing complex patterns in delta smelt otolith chemistry. And so we collaborated with IEP to get an archive of over 2,000 wild-caught delta smelt across nearly 20 years from the majority of the spatial extent of the species range, as well as environmental variables in the delta over that time period. We categorized those fish into 3 different life histories: freshwater residents, where the entire profile was below 0.5 salinity; brackish water residents, where the entire profile was above 0.5 salinity; and migrants, which showed a transition from fresh to brackish water. And when we look at how those profiles change over time, we can see that these life histories are persistent across years. They're not just showing up in one year or another year, but they're also highly variable from year to year. And so we can ask, how does this variability, how does the proportion of these life histories in any given year change in response to environmental variables, in particular mean summer temperature and mean freshwater outflow? And what we found is that both temperature and outflow were significantly associated with changes in the proportion. In particular, cool, low flow years were associated with increased proportions of freshwater residents, whereas warm, high flow years were associated with increased proportions of brackish water residents. The migrant phenotype was the dominant phenotype, and across all conditions, it was generally relatively abundant. So in summary, we can say that delta smelt are exhibiting complex life histories that the otolith microchemistry is doing a good job of capturing, that those life histories are persistent through time, and that they're strongly influenced by climate. And while it hasn't been assessed, it's possible this life history diversity has impacts for the resilience of the overall population, which is something to keep in mind as we start doing introductions of cultured fish back into the ecosystem. And with that, I will finish. And I'd like to say my PI, Levi Lewis, is giving a talk on Thursday in the longfin smelt section. And I encourage everyone to check out his work for more of what our lab has been doing.
Thank you. Our next speaker will be El Patullo from the University of California at Davis.
Hello everyone, thank you so much for attending my lightning talk today. My name is Caitlin Patullo, but feel free to call me Elle as all my colleagues do. I am currently a master's student at the University of California, Davis, and I do some really interesting research in the Conan lab that I am so excited to share with you today. As you can see, the title of my talk is Drug-Induced Hypo and Hyperactivity in Larval Longfin Smelt. So my research focuses on stress-related behavioral changes in early life stage locally threatened fish species. However, the study in particular is hoping to further understand the effects of hyper and hypoactive states on behavioral changes using two pharmaceutical drugs. The first is pentylene tetrazole, which will induce a hyperactive state, and the second is midazolam, which will induce a hypoactive state. And our primary behavior of interest is photomotor response, which is essentially the fish's reaction to changes in light conditions. However, we're also working on developing a predator avoidance assay using alarm pheromones or other predator-related olfactory cues as olfactory stimuli. The information that we will gather from inducing these two behavioral states will not only allow us to better understand the nature of the stress response in local fish species, but will also allow us to establish positive controls for future studies regarding contaminant exposure, which is extremely useful for us to have moving forward. So this slide outlines our experimental design for our photomotor assay, which is our best established protocol at the moment. In this protocol, we expose our larvae to 2 light photoperiods, which serve as our stress-inducing stimuli, as well as 3 dark periods. The first acts as an acclimation, and the latter 2 are used to help us better understand and look more closely at the larvae's ability to recover from the stress of the light conditions. We also use a Noldus Daniel Vision chamber to film our trials, which provides us with the controlled environment to conduct our experiments. And we use ethovision to program our photoperiods and track the larvae's movement throughout the experiment, which yields an output like this. Nice and clear picture. Now the fun part, our findings. This graph is one of the many graphs we've acquired from our pentylene tetrazole exposure assays. The first thing I should point out is that the peaks that you can see from the 10 to 15 and the 20 to 25 minute timestamps are light periods. So we're able to observe an increase in activity in response to the light condition as we expected. Second, it is important to note that the blue line in this graph is our control group, which displays the lowest amount of distance moved compared to our experimental groups in all of the photoperiods following the initial acclimation period. This shows us that PTZ is effectively inducing a hyperactive state in these fish, regardless of the light condition that they're exposed to. And this graph is from one of our midazolam exposures. Similar to the previous graph, the peaks that we see from 10 to 15 and the 20 to 25 timestamps represent our light periods. And again, our control doses are in blue, with the solvent control dose containing less than 0.1% of methanol. And what is special about this graph is that throughout the entire experiment, we can see that the doses of midazolam result in lower amount of total distance traveled, regardless of whether the fish are experiencing light or dark photoperiods. This suggests that midazolam is effectively inducing hypoactivity, which is a thoroughly satisfying and very exciting result as well. Okay, and that is all I have for you today. Thank you so much for joining me for this little talk. And I want to quickly, before I go, thank everyone who helped with this project. As you can tell, there have been numerous individuals in the Conan Lab that have contributed significantly. And we've also gotten some guidance from a few of the personnel from the Lion Lab here at UC Davis as well. And yeah, none of this could have been possible without them. And we're very excited to see how the results turn out in the next couple of months. But yeah, thank you so much for joining again, and I hope you enjoyed the rest of the workshop. Bye.
All right. Thank you, everyone. If you're still here, if you just gave a lightning talk, go ahead and turn on your camera. Maybe people can call up the gallery Everyone wave to the crowd, say hi, and thank you. That's the end of the lightning talks. We're going to transition over toward the open mic poetry slam portion. So thank you all. I know my head is bursting right now, but I love that format. So I hope you all did too. Thanks everybody for being flexible and trying this format out. Give us feedback if you think this was worthwhile. But thanks all of you. That was really wonderful. A really good overview of incredible research that you all are doing. And I appreciate that you're sharing it with us today at the IEP workshop. We've got a couple of scheduled speakers for the Poetry Slam. This is, I think, best characterized as our open mic session. A couple of them came to us identified, and we'll move right into those. And we have until about 4:45, I think, 4:30, 4:45, you know, it's flexible. Christine, do we want people to start? We'll do these two talks that we have now, and then we'll raise hands. Is that how we'd like to do that?
Yeah, I think if you have them raise their hands, then we can elevate them to the panelist level and they can turn their camera and their microphone on.
Okay, yeah, I don't see those. So I guess you're gonna have to keep track of that. And maybe you can let me know once we finish the next two talks, which we know about, how many we've got, and then that'll help us determine sort of how long we can go, how many people are interested. But to introduce the rest of the session, our first poetry slam open mic participant is Rhiannon Mulligan. Rhiannon.
I don't think we saw her, Steve. She never came up to the panelist level and we haven't seen her in the attendee level.
Okay, then we'll move right along to Brandon Lehman.
Great, that's a pre-recorded video. I will share it now because
okay, and I would encourage those of you who have Poetry Slam presentations to go ahead and queue up. We'll start raising hands now.
Hi, I'm Brendan Lehman with the National Marine Fisheries Service and the University of California, Santa Cruz. This one's called Blinded by the Light. Blinded by the light, eaten by a bass in the middle of the night. Blinded by the light, eaten by a bass in the middle of the night. You see these banks along the levees full of human activity, illuminates the midnight sky, and the salmon on their way to the Golden Gate, distracted by the lights as they swim by. Docks and piers and the street lamps and the bridges need to be lit up so we can see. But maybe there's a way we can reduce illumination and let the fishes swim around in peace. Blinded by the light, eaten by a bass in the middle of the night. Blinded by the lights, eaten by a bass in the middle of the night. It's a bona fide feeding frenzy, native fish buffet. First thing that we had to do was survey all the habitat with a light meter on our boat, take a drive at night through all the sloughs. Headed down the levee with our clipboards at the ready to document everywhere that's bright. And now we have an inventory, slightly more complete story of everywhere in the delta that there's lights. Blinded by the light, eaten by a bass in the middle of the night. Blinded by the light, eaten by a bass in the middle of the night.
Woo! Nicely done. All right, off to a winging start. Christine, who's next? Do I see Levi Lewis?
Yes, I believe so.
Levi.
Hi, good afternoon. Can you hear me?
Yes, I can.
So I'm Levi Lewis. I'm the PI of the Otolith Geochemistry and Fish Ecology Lab at UC Davis. And I would just like to share a quick poem that I wrote about our favorite stone. I'm going to try to share my screen real quick so you can read along.
Yeah, we're good.
I quietly accrete, silent and discreet, document with element life histories complete. A stone that never rolls, vibrate, communicate, serving many roles, orient, accelerate. Quite often I find, from skulls I'm often mined, like a diamond in the mind. Hosted dead, so doesn't mind. And the scientists, they smile as the run files compile, knowledge growing all the while, with hypotheses on trial. Thanks.
All right. Thank you, Levi. So where are we? Any other takers?
Not yet.
This is the point where everyone gets a little embarrassed and they're pushing their neighbors. Come on, guys. Any other takers today? I know it's been a long day.
So far, not yet. What about you, Steve? You have anything you can ad lib for us while we're waiting for hands?
Not a good idea. Not a good idea. So while we wait to see if there are any other raised hands, let me go through a couple things. So the poster session, I wanted to give thanks to the organizers, Emily Risnar from the Delta Science Program and California Sea Grant. I wanted to say as well, thanks to Michelle Avila from CDFW. The poster session is designed to host your visits and questions and interactions from between 4:45 and 5:45. And then, of course, we'll reconvene and begin additional workshop sessions, session 5, tomorrow morning at 8:30. Once the workshop is finished and we figure out how to do it, the workshop recordings will be available online until the end of June. In case you missed anything today or have conflicts, we will be posting the talks for a couple of months. You'll be able to find them online. And don't forget to vote for the best early career posters. There's a link in the materials we provided so that you can do that. And then there's also a people's choice for you to vote on what you feel is the best presentation. We will acknowledge that at the close of the workshop or very soon after. You can vote until Friday, until 5 p.m. We'll announce the winners next week. Anybody else, any takers for the open mic portion? I know this is a little weird that we're doing this virtually, but thank you to our two participants.
Yeah, nothing yet, Steve.
Okay.
Oh, I spoke too soon. We have someone willing.
Oh, yay. All right.
Let's Janice, we're going to bring you up. We're going to have Janice Cook from the Central Valley Regional Water Quality Control Board. So Janice, when you are, we've elevated you to the panelist level, feel free to unmute and share your screen. There you go.
Okay, this is not, oops, there we go. Nothing like the previous two, but a little haiku about looking forward to Delta in June. Warm, shall I swim? Ick, habs are so depressing. I bike instead.
Thank you.
We need one
Janice.
of those applause things on here, you know?
Yeah, we need the team's applause cues. All right, well, I can stand uncomfortably for another minute or so, and then I think we'll call it. If there are going to be any takers, speak now or forever hold your peace. So if not, Christine. Anything we need to say about the poster session?
Yeah, just hopefully everybody will sign back on around 4:45. It's a Zoom meeting versus a Zoom webinar. So if you're familiar with Teams and how we do meetings in Teams, it'll be very similar to that format. We're going to have breakout rooms based on the categories of the posters. So hopefully all of you have had a chance to check out our poster discussion forum to see how the various posters are, categorized, like Delta smelt, longfin smelt is a category, salmon is a category, water quality is a category. So anyway, we're going to have breakout rooms with those poster presenters with that category in the room. And then basically they're going to run their own meeting. This is an opportunity for you, just as if you were milling around in the little room at the Lake Natoma Inn, checking out people's posters and asking them questions. This is going to be a very similar type of format, except it'll be more of a group effort based on, again, the category of the poster session itself. So we're hoping that people come back and that you interact and participate and ask questions to the poster authors. I know that they're going to be available today and tomorrow. And again, also check out our poster forum. You're welcome to create an account and leave a comment for the poster author in the chance that you're not able to engage with them today and tomorrow.
And those relevant links are online on the IEP workshop page. Is that correct?
Yeah, so if you're talking about the link to the Zoom meeting for the evening poster session, yes, those are on the website. They're also in the program. And if you're talking about the link to the poster discussion forum, yes, that's also on the website, which is on the annual workshop webpage.
Okay. Anything else?
Nothing that I have, and still no takers.
All right. Well, I think I'll spare us any further uncomfortableness, at least mine, and say good day all. See you at the poster session, and see you all bright and early again tomorrow. Thanks a lot for your participation. I hope you've enjoyed the day as much as I have. So long, everybody.
Bye. Thank you.